Nice to meet you. My name's Nico.
Yeah, good to meet you, Nico.
Yeah, thanks a lot for taking the time on my call. So, yeah, I saw your post about tech transfer challenges, and so yeah, that's something that I wanted to talk to you about.
It's hopefully a thing of my past a little bit, but I've worked in CDMOs and I hired CDMOs when I was on the other side of the bench as well. And I talked to a lot of people. I know a ton of people in the industry working in CDMOs or that use CDMOs. And cell and gene therapy specifically, but I think all biotech has the same problem, that all the science has a bit of nuance to it. There's a bit of art to some of the things we do.
Yes.
In cell and gene therapy, more art than maybe in other areas. But even in general monoclonal antibody production, it can be the same. There can still be a little nuance to that is hard to transfer over. And I think the issues we run into, like I put in the post, is people come and because of especially investor pressures in our space to get and move quickly, everything's done subpar, right? They're done in a rush. They're done without the preparedness required. And it's inevitable that there's going to be a train wreck somewhere in there.
Yeah.
I do think if people take their time and walk through it slowly and make sure that they are really documenting really well, there's a good handoff, it can be successful most of the time, right? It can be a really good rate of success. There's always going to be some challenges just because of the fact that science is moving site to site. And even within the same company, they can have difficulty moving things site to site. There's just enough challenge in doing that. You move it to a totally new team that is unfamiliar with it, you're going to have some failures. But we just tend to increase the failures due to our own shortcomings, I guess you'd say, usually due to the need for speed. And can't say how many times we've heard it. We've got to hit this timeline, and their timeline is just unrealistic, right? And we just aren't willing to budge on timeline, even though you tell them that the odds of failure increase exponentially by trying to short circuit a normal process. Because usually on the CDMO side, they have a pretty set process what they want to see, right? X amount of time here, X amount of time here, X number of runs, all that kind of stuff. And as soon as you try to short circuit that, you ratchet up your chance of failure significantly. And I can't think of a time where that failed, where we actually made things move faster than they were supposed to and ended up in failure. I don't think there was ever success that I saw in doing that.
So I heard engineering runs. I hear this is one of those things that you're talking about that sometimes gets brushed to the side for the need for speed.
Yeah. And it's a combination of speed and cost, right? People think you're just trying to milk them for cost, right? Oh, why do we spend another $100,000 on doing that and all the materials and the time it takes? A lot of money gets wrapped up in that. You could spend $1 million on engineering runs easy, even in cell and gene therapy on autologous process. And they don't see the need for it. But the need for it is there's a lot of things to iron out, even from having successfully done the process in R&D to successfully completing a GMP run. They want to try to bypass that entire middle ground of making it work in the GMP, documentation correct, BOM correct, people trained, documentation fully vetted, and QA oversight. All those pieces have to happen, and they want to basically just shrink it to nothing, right? Unfortunately, that's where a lot of people are, and I think that's one reason CDMOs have a bad rep. There's a lot of CDMOs that are good. Most of them are willing to short circuit to get the contract. They usually, you won't turn down a contract because they want to do that. I have, right? On the BD side, I basically said, we're not going to do what you want to do. You have to go someplace else. Because it was so extreme. But usually you can come to an agreement with, and no matter what kind of agreement you put in place, they always blame you when it's done. If it fails, they blame. But if you aren't willing to put the time and money into it, you kind of have to expect that.
Yeah. And that makes a lot of sense. So something that's come up in a lot of these conversations is technical risk, because this really is a technical risk management game we're talking about. And one thing I'd be curious to hear your perspective on. So cell and gene, this is a not immediately new, but still pretty new technology. And one of the consequences of new technologies is they're less characterized. So they are less understood how they operate at the large scale than, say, older techniques for manufacturing and older methods. So I'd be interested to hear your perspective. Do you see differences in these tech transfer challenges with new technologies like cell and gene? Do you see more of these communication errors because they're less characterized?
I think they're less characterized, which hurts, and I think there's less platform technology, which really hurts. Whenever you're learning a process in monoclonal antibodies, you're still doing probably 90% of the stuff the same, right? In the way you grow the cell line, the way you crack it open, all that kind of stuff. There's so many similarities across it because most of these things are very platform, right? In cell and gene therapy, there just isn't that for the most part. And we need to get there. But that's one of the biggest problems. So when you get a new process, you're getting new technology, you're getting new everything. It's like you're building everything up from the ground. And that's, if you had an architecture that covered 75% of it, and you just had to build the 25%, it'd make it a lot easier. And that's typically what they do in the mAb world. In cell and gene therapy, you kind of start from ground zero every time. And
everyone has their own infrastructure, basically.
Yeah, every single one of them. And even if you do 2 CAR T programs, we did 2 CAR T programs at the exact same time at one place I was at. They were so different. My goodness, you couldn't have been. One was lentivirus, one was a transposon, right? One was a really long period. One was trying to get down to a 6-day. The other one was like a 14-day. The bomb was like 30,000. The other one, the bomb was 200,000. They were just night and day. And the processing, the number of steps and the number of automations versus manual, they were just totally different as far as you can get apart. And so you think bringing on the next CAR T is going to be easy, but it's starting over from scratch. So yeah, the technological risk is there largely because you're always starting over, right? You can't bring anything that you already did before to bear. Those learnings are almost irrelevant sometimes when you go to a new process. You try to, and the more you do it, the more programs you bring under, right? The more that you get, but the base builds very slowly, as opposed to some other technologies where that base is already really high and you're just having to top off the little bit of knowledge.
It's foundational at this point.
It is.
Yeah. So something else that I'd be interested to hear your perspective on, specifically in this tech transfer. So with antibodies, say for vaccines and things like this, it's a bit different from my perspective compared to personalized medicine like cell and gene. And that difference being that if you make a mistake when you're manufacturing something generic, as long as you cache it in quality control, you can throw that batch out and you'll lose some money, but you can just do it again. Whereas there is a scarcity of patient cells here that I think it creates a different it increases the risk profile to some extent because you have a lot less leeway to get that right.
Yeah, definitely. The financial risk in some way is lower because you're doing one run at a smaller scale and things like that. So if you mess it up, it may not be a $3 million batch like it is in antibodies. It may be a $200,000 batch. But like you said, the starting sample may be a one-off, right? You may never be able to get that again. So when you go to allogeneic therapy, you're limiting that risk a little bit where you can get normal donors and you can get a sample back and you get more into that larger scale, larger cost, but less risk on the front end. But yeah, having a scarcity of sample on autologous is a huge factor. And the other one is in all these other ones, you have downstream steps that can clear things for you, bioclearance steps and all this kind of stuff, right? You're biosafety conscious, you're not sterile. In cell therapy, different game. You can't have anything happen. It has got to be sterile from kit to caboodle. And if you mess up anything in the middle, you throw out the batch. So the risk profile on the manufacturing side goes up another notch. And we don't have any safety nets on the downstream side.
Yeah, that makes sense. That with other processes, if you introduce a contaminant, you can filter it out, but you don't have that opportunity here.
Right.
Yeah, that it makes a lot of sense. I'm curious to hear how you see documentation playing a role in this sort of risk profile of these technology transfers. Do you see that being a big part of it? I know tech transfer, there's legal aspects, there's the actual operational handoff, there's a lot of different parts. So I'd be interested to hear if you have anything to say about that aspect.
Yeah, I think this documentation comes up in 2 ways. One is kind of that preparedness, right? How thorough are your documents going in? How good is the instruction set going to the operators? Because most of them, you're going to have PhD people in there typically. You've got people with associate's degrees doing this, which are good. They've got good hands often, but they're not going to be discovering on their own. If there's a problem, oftentimes it's an impasse, right? They're looking at instructions, they're following instructions. It's like building a car, right? Okay, if it says to do A and B and part C is not available, what do I do? And it's the same way in the lab. You're not having R&D scientists doing that. They can do a bush fix, right? They can't work around an issue like that. So the documentation going into manufacturing has got to be rock solid and got to be thorough and got to be to the point where if things do come up, it's flexible enough they still have a path forward. They don't get stuck in their tracks. The other part of that is the documentation that is going into what they're doing and how challenging that is, which is why I love like electronic batch record, things like that, because they do 2 things. One is they make sure you don't go to step A before you've done don't go to step B before you've done step A, right? They sequentially walk through you things. You can't skip things. You can't misalign things. But the other one is a lot of the errors we see in documentation are illegibility, they forgot to do something, they did a transcription error, really small stuff, but boy, that can really screw up everything, right? From the point of, was it really done correctly, not knowing for sure, because you put something in the wrong spot. Does that mean you actually did it in the wrong spot? Or did you just write it in the wrong spot? To the point of just delays in all of the QA oversight of trying to figure out, okay, we've got to fix it because they wrote their initials in the wrong spot or they wrote the wrong date, and fixing all that kind of stuff can just take a long time. And disposition of these products just drags on and on. We were working to get our disposition time under 60 days at one place. And at another place where we had an EBR, our disposition time we were trying to get done in 48 hours. Right.
Wow.
That's the power.
So the digitization makes a drastic difference.
It's just night and day, right? Because you can do everything in real time when you have EBRs. Everything is done. The only thing you have to look at is if you had a true deviation where you have to go do an investigation, that'll delay things. But if not, everything's done in real time. And at the end of it, typically we had one product disposition 20 minutes after sterility came on, right? Because everything was done. All they had to do was hit the button. Even in real-time monitoring, when you have humans doing all your QA, 1,000-page batch record, it's still going to be weeks after the process before you're actually ready to go. So I think it's a huge efficiency thing, but it's just, again, that safety net, right? Digital people don't go digital stuff. It doesn't get tired. It doesn't lose track of where they're at, all that kind of stuff. People do. And I think having digitization as a safety net in this space where there's so much detail is really critical. And now that we have the capability of doing it, there's no reason we shouldn't be implementing it more. Yeah.
So digitization definitely seems to be the current trend. And it also seems to be a way to capture some of these details something that's different about these procedures is they're very sensitive. So you can't have 99%, following 99% of the SOP isn't enough. You need to have 100% of that SOP followed correctly. And something else that I've heard through conversations that is a challenge in these tech transfers is that the standards for documentation between R&D and process development are generally different. So people working in R&D often are PhDs or come from an academic, like a research background where the standards for reproducibility might be like N of having 3 replicates, for instance. And so that sort of culture creates an expectation that, okay, once I've done the 3, I'll write it down, sign it off, and that's it. And then when engineers receive that, there are often these cracks. Yeah. Do you see this in your experience?
Yeah, I think there's actually a third level. So you get R&D that's definitely very lax. And I worked in R&D for a long time, and I kept my lab notebook on a folded piece of paper in my coat, right? Bad training, but it's typical, right? You move to PD, people that are much more kind of putting in process robustness and working on trying to GMP as a process. They're much more dedicated about documentation, I think, and stuff like that, especially because they're working on things that they may go to patents and all this kind of stuff. And it's drilled into a little bit more. And they're also the ones oftentimes starting to make your batch record and stuff like that. But even they are not to the degree of what has to happen in the GMP, right? Where you have verification signatures, everything's got to be dated and initialed in a certain manner and all that kind of stuff. So there's 3 different levels, and you have to make sure that when you move from R&D to PD, you don't lose things in that transition. And then when you go from PD to GMP, you don't lose things in that transition. Having to go through multiple transitions like that and having people talk different languages along the way, it certainly adds to the level of complexity and the level of the challenge in compliance, I guess you'd say, once you get to that ultimate goal of GMP.
Yeah. And I've also heard that once something is working at a later level, that no one will ever want to go back because the bureaucratic challenge of getting through it again is such a high barrier that there's a lot of pressure not for that to happen.
It's true, and there's a lot of pressure not to make changes, and there's a lot of reasons for that. A lot of it is regulatory too, right? You don't want to make changes in processes because then are we starting over? Do we have a new product now? All that kind of stuff. But yeah, part of it is just the burden of compliance in doing this kind of stuff, getting batch records ready, training people to do it, all that kind of stuff. As soon as you make a change, you've got to start some of that over, right? Not all of it, but whatever you change has got to be now reset. And it is difficult. So you do want to make it's kind of like you want to make sure everything is right before you go in, so you don't have those changes. But the problem is you do learn along the way, right? And so you have to have a system. And again, this is where EBRs are really nice, right? Because you can make iterative changes much faster, and you can have that lineage of knowing exactly what changed when. And in a paper system, so often you lose that. It's like, okay, we had this 4 years ago, we have this today. I can't remember what's in the middle, kind of what
Yeah,
got
yeah,
meshed.
yeah.
But you have all these, all your transitions are documented typically in electronic records, stuff like that, where you can go back and say version control is pretty tight in that.
Yes.
Because in a paper system, version control, we try to keep it tight, but I would say it's not always great.
Yeah, yeah.
Um,
It makes
so-
a lot. Yeah, it makes a lot of sense. And so I'm not sure if this is the same in the U.S. In Canada, I'm hearing that there's a lot. It's a slow adoption process towards electronic batch records. And one of the sort of perspectives I'm hearing is that it is a bit of technical risk. So for a company to implement a big change in their documentation practice, that is a big change. You have to train everyone, and that's a pretty big cost. But if you think about it down the line, the risk mitigation in terms of having access to all of those change logs and all of those details that are actually accessible might be worth it. So I'd be curious to hear your perspective on that.
Yeah, I will say before the last CDMO I worked for, I thought batch record EBRs were much more meant for late stage work, right? At least phase 2, if not later, because of the difficulty with implementation and the cost. And there is a huge upfront cost. Like CDMOs, there's a big upfront cost. And it's also a change from a legacy system, like a paper-based system, is very difficult, right? How do you transition all the stuff that's paper-based into this and all that? That's hard, right? So starting with a new CDMO and starting with the electronic batch record was nice. It was costly. But what I learned was that when you build a system that's suitable, you can do iterative changes at EBR even easier than a paper system, right? Because how easy is it to keystrokes, right? And
Yeah.
you can document all that kind of stuff. It was actually, the transition was much easier than I expected it to be. And the version control made it very easy to move and iterate. Most of our programming was done overseas too. So we could actually tell people, okay, this is what we want changed. And while we were home sleeping, they would make all the changes. You come in the next day and it's all done. So it was pretty easy to go through iterative changes, actually easier than it was in a paper record. I think in the long run, the biggest thing is cost, right? It's expensive to get into. It is a challenge on training and all that. All the cost that goes into the transition is real. And it's daunting especially for companies that have legacy paper systems that are rooms of file cabinets. It's daunting to figure out how you're going to transition that into something else. So I think it's something we have to move toward. I do think after seeing it real world firsthand implementation, it's such a lifesaver in the long run. And the other problem is, especially for CGT, there's not really good off the shelf products. Everything needs pretty much custom fit. And because, like we talked about in the beginning, you have so many process changes, your EBRs have to be built from scratch almost every time. Just like the process itself, you're having to build the EBR every time. You can't have a platform that says, okay, 75% of it's set, we just need to make these few changes. It's, your process changes, so your EBR changes, the whole thing. So that there's a delay in getting started sometimes, and that's a real delay too for clients that they've got to understand. I don't know if it's too much more in the EBR than it is in paper, but typically you're still doing the paper first and transitioning it to electronics, so there's a little bit more delay maybe. But I think that is very much outweighed by the benefits that you get from the EBR in the long run.
Okay. And I'm curious, so those, like you mentioned that you have to have a separate EBR for each kind of run with cell and gene. So why is there a reason that's different with cell and gene compared to other pharmaceutical products?
It's just a different EBR for every process because every process is so different, right? It's the same EBR, but you're building each batch record from ground up, right? You don't have a template that you can use as the basis and then just make changes to, because there's so many differences in the inputs and in the processes that you might as well just start from the beginning every time, right? You could maybe build a template that you could try to shuffle, but the way these systems work, I think it would take longer to do that than to just start over. Where in a monoclonal antibody, if 75 or 80% of your process is the same, tweaking that last 20%, that's easy. So having a template that you manipulate a little bit is fine. And eventually in cell therapy, we'll get there, right? Gene therapy, probably a little easier. The vector batches get much more like batch manufacturing, like monoclonals. In the cell therapy game, though, there's still so many different processes. I think the more mature the industry gets, the more platforms we'll see, and the easier it'll be to get into templates in there as well. I just don't think we're there yet.
Okay. Yeah. So because there are so many different processes that people have to use in this space, that's what really makes it different. Whereas with the antibodies, you can have the first 3/4 of the process the same and then tweak downstream
Right.
a bit for purification of a certain type. Okay.
Yep.
That makes a lot of sense. Yeah. And so I'd be curious to jump back to the documentation in the tech transfer of the methods there. I'm curious if you see an opportunity for improvements in capturing like process specific details. You mentioned that the people at, my understanding was the CDMOs might have like a associate's degree or might not necessarily have the ability to do in the moment troubleshooting that someone who developed the process might. And I'm curious if you see an opportunity to improve the detail capture of the person that developed the process at the, say, the sending facility, to give some flexibility for that person at the receiving plant to not get stuck in their tracks, so to speak.
I'm sure there is. I don't know that I'm smart enough to figure that out, but yeah, and I will say I've been looking at AI a lot more, and I do think this is an area where AI could help. I think having electronic records where you could have AI look at a situation you've gotten into and help you find the best path out would be an option here. I've been pretty impressed. I was very skeptical of AI even to the end of the year. I've just started looking at a lot more and doing a lot of kind of AI discussions and challenging it. And I've been amazingly impressed by how accurate it typically is, and the kind of thinking the outputs are typically very well thought out and better than what a lot of people would do in their outright, especially in the timeline they do it. It comes almost instantaneously, right? If there's a way to get somehow some sort of AI kind of capability in these things where there could be a help that way, that would be a great idea. And I think it's probably possible these days. So the regulatory ones, I don't know, blame it on AI. There's something to do. That might be an issue. I don't know how we get around that part, but I certainly think that kind of that quick recall of here's the options available to you, and here's the most likely scenario that's going to be successful, seems like there's got to be a way to make that work.